We will continue with a PhD student from the laboratory of genetics, Theeuwen, and he will tell something more about a very basic process, which is photosynthesis.
Indeed. So, good afternoon, everybody. My name is Theeuwen. I'm a PhD candidate in the laboratory of genetics and supervised by Mark Aarts. And the next 10 minutes or so, I'm going to give you an insight in the evolution of photosynthesis, which is basically a 2 billion year collaboration. And so, let's start at the beginning. Here you see a very eukaryotic cell, a bit further along the line of evolution than Daniel was describing before, and a free-living photosynthesizing bacteria. Now, these two ended up collaborating, or the eukaryotic cell engulfed the photosynthetic bacterium, and this ended up being the organelles and a separate form, that's the nucleus. And this symbiosis, or this collaboration, paved the way to plants as we know them nowadays. So if we consider this as a plant cell, and we zoom into this, then we see the following. So we see the two organelles and the nucleus, and we see that these three all have genes located on them. And for me, interestingly, is that the chloroplast at this moment has roughly 85 genes located on it. This, while the early photosynthesizing bacteria, its ancestor, had close to 3,000 genes. And those 3,000 genes either have been lost or moved to the nucleus during the course of evolution. And this is actually what we see, because the nucleus has much more, like 27,000 genes in this particular case. So this means that this nucleus is really strongly determining everything that's going on, and there are these two organelles still playing around here. Okay, so, but the process of photosynthesis is actually still playing on inside the chloroplast. And photosynthesis is very, very complex. It requires up to 2,000 genes to properly function. But I said there are only 85 genes inside the chloroplast. So this means that there are actually a lot of genes, or products of genes, coming from the nucleus into the organelles. And to make stuff even more complicated, there thus needs to be a feedback loop as well, because if there's something changing in the light, as we've just seen, the chloroplast needs to tell the nucleus, okay, something changed, give me something else. So, of course, there's also this feedback loop. So what you already can see here is this a very tight, complex interaction. And I'm particularly interested in breaking this apart. So before I'm going to tell you how to do this, there's something else that I need to explain you. And that is basically what is natural variation. So just like us humans, none of us is the same, and the same is true within a plant species. So this variation is actually manifested in the form of genes. And as I already told you, these genes are actually located not only in the nucleus, but also in the organelles. So if I show you those two plants here, it would be ideal if we would be able to actually change the organelles freely among plants. And this is something that, or the resulting plant of this, is what I call a cyclic. So how does this look like in theory? So what you see here is 5 different nuclei with their own wild type, hence the WT, organelles. And they just look happily like this. But what if we were able to give them the set of organelles of a completely different plant? In this case, plant X. We might see, or the result might be, that they are actually becoming much bigger, all of them, regardless of the nucleus. Another option would be that in combination with the organelles of plant Y, that one of them only got bigger, a couple of them would stay the same, and one of them became smaller. Hence, this is the resulting outcome, the phenotypic outcome, is very much dependent on the specific combination between the organelles and the nucleus. Okay, this is a theory, so how can we do this in practice? So here you see 2 plants again, and these plants are, and if I would schematically show them here, the following. So the blue one is the female parent, and the orange one is the male parent. And by schematically I mean the following. So on the right hand side you see those 2 stripes, they represent the chromosomes and inside the nucleus. and the round circle around it is representing the cytoplasm with organelles inside. So hence blue and orange colors there. So there's something else interesting in here, and this is actually making my work possible, is that we make use of a mutant line. So this female one is not a normal line, but it's a mutant line. And I show this with these 2 green dots. And before explaining you what it does, you will see later, but it's important to know. So we just cross these 2 lines, and then the following happens. This is very typical, so 50% of the material in the nucleus is coming from the mother, and another 50% is coming from the father. Hence you see this combination of blue and orange. Now the organelles are only coming from the mother, and that's very important. So here we have already an offspring, but I said there is also the mutant phenotype present. So you see this green dot, and actually what it does, right after fertilization, this part is completely lost from the offspring. So this is what we call maternal genome loss. And so at this moment we have an offspring that is only a haploid, so only 50% is still there, but it's only consistent of the nucleus of the father and the organelles of the mother. So one more generation of selfing creates a double haploid, and this is the sirene, a perfect nucleus from the father and the organelles of the mother. So we did this, and we did this for we gave 7 different nuclei the same set of organelles, but also we made all other combinations possible. And for the really quick ones amongst us here in the diagonal, these are actually we remade those the original ones, and these were actually there to make sure that our mode of doing this, the mutants didn't actually interfere with this phenotype. So they were there as a control. So, like I said, I'm trying to study photosynthesis, but just looking at plants, it's quite hard to do actually, to look whether one plant is doing better in photosynthesis or worse. And you can see here a bunch of plants growing there. Some you might actually spot are a bit lighter green than others, and this might be linked to photosynthesis performance, but I can tell you it doesn't necessarily need to be. So that's why we have this robot, we call it a phenovator, and I have it shown here in a little image, and what it does here is it sheds a certain light regime onto the plants, and then measures amount of light being reflected again. And in doing so, we can, using a couple of different of these light regimes, we can actually extract a lot of different photosynthetic parameters. Okay, so we do this, and we can do this for a long time, and the outcome is actually something like this. So this is a false color image of a certain trait, in this case photoprotection, and what you see here is actually on the horizontal level, the top 2 ones have the same nucleus. It's falling off a little bit, I see, that one, but why not? So the top 2 are having the same nucleus, but having different organelles. And the same for the bottom 2, the same nucleus, but 2 different organelles. And you see that this trait, in red, I show that it's worse at doing photoprotection, and this is actually linked to 1 nucleus. So actually this trait is linked to a nucleus. But if we look at a different trait, in this case photosynthesis efficiency, it has actually something different going on. You see that the orange red part is showing worse performance, but this is actually linked to the organelles. And you see that in this case, the stripes are going vertically. And with this new technique, we are able to make sure whether a trait is caused by the genes located in the nucleus or in the organelles. So this is a fascinating thing which we were not able to do before. And if I now do this a bit more quantitatively and show this in a graph, like here, you see that this line, the dots on this line, are actually the same nucleus, but with different organelles. And you can see already that one of them is actually doing much better, and one of them is doing much worse. And if I throw in the rest now, you can see this is a bit of a mix-up, but what you see now is that there's one set of organelles, regardless of the nucleus, causes a 20% decrease on average. But there's also this one here, that regardless of the nucleus, increases photosynthesis efficiency by 6%. So this variation that we now see is something that we were not able to see before if we didn't specifically separate the organelles from the nucleus. Then it would just be the nucleus determining everything, if you will. And now we're actually doing so. So, and as I already said so, by doing so, we are revealing a hidden form of variation that we were not able to do so before. So if we then look at this image with some of our crops and a huge amount of variation that is there and that we're actually using to breed our crops even better, we could question ourselves, what else is there? What more hidden variation is actually present? So to sum up, my question is, what else is there? Thank you very much.
Thank you, Tom. Excellent work. What else? Questions? So what determines then the interaction between the chloroplast and the nucleus? So what is that interaction that gives a positive final output in terms of photosynthesis?
Very good question. So in this case, I'm showing an example. This is only one trait, but we measured more than 1,500 traits. But in this case, it's very clear. You see that regardless of what the nucleus is doing or what the genes are in the nucleus, this phenotype is there. But so in this case, is determined by the genes or the alleles of those genes in the chloroplast in this case. But of course, it can also be a different traits show that it don't show this clear-cut difference, that it depends a lot on the genes or the alleles of the chloroplast in combination with certain alleles of the nucleus. So it's very much like if only the two meet, then there is the phenotype present. So it's a very tight, complex interaction. And we are using natural variation for this, and that's great because we don't have to do anything, it's already there. But we are tied on seeing what is causing it. So we need to have huge panels to actually pinpoint a certain gene causing this. And this is something I'm doing now. So.
So you show this 6% increase and 20% decrease. Is this a general pattern that it's harder to get a gain than to get a loss?
I think you can be, yes, I think in general it's not easier to break down something than to improve something. But I can also tell you this 20% this decrease was something that was used as a sort of internal control. So we knew that this one was doing much worse. So in a sense it is great that it's there, but we already knew it. The other one that's showing the 6% increase, we had no clue it was there. So in this case, and I'm focusing on photosynthesis now, but there are many more traits, but we do see spread over different set of organelles, there are increases more over the place. So it's not the only case by any means. And this is not only the only trait that we are seeing it, because this is linked to many other traits. A very, very, very bad question.
Do you see the same trend when the plant is bigger? Because here you're doing the experiment with the small plants, and when the plant is bigger, in the last stages, for example, producing food, do you see the same trend, or is it going to change?
A good question. So I left this a bit in the blank, but I'm using a whole species the same as Vera was this morning using, and this, as we are measuring, as we saw it in one of these images, is already the big stage. Okay, it's not flowering yet, so not producing any fruits, but this is the stage in the mature stage, if you will, and thus we do see this in the mature stage. Absolutely, this is not a crop that we can eat, so this is some of the research. It has to be transformed into a more applied side, and then we can test this. But in general, the fact that we see this, I mean, photosynthesis is something in the beginning. If it's producing a bit more sugars in the beginning of life or all the way up, it's basically exponentially going. So if there's something in the beginning, we can predict there's something else in the end as well. So.
Okay, thank you.
No more questions. Thank you again.